Hello everyone, welcome to my YouTube channel. Today I'll be presenting my final project for Biochemistry 2 at Geneva College, taught by Dr. Rodney Austin. I hope you'll enjoy this video and perhaps learn a little something from it as well. But before I begin, I'd like to point a little bit toward a little biblical connection. Psalm 113:3 says, "From the rising of the sun to the place where it sets, the name of the Lord is to be praised. All of creation attests to the glory of God." At many points in Scripture we see people attesting the splendor of the sun and the wonders of creation to God's glory. In many ways, by having a more informed view of how that creation works, we can better see how much glory is densely packed into even the smallest things in our world. I hope that this presentation will reveal to you a little bit of that amazing glory to be seen in every blade of grass. Today I'm going to be talking about some of the basics of photosynthesis from a biochemistry standpoint. We have known for a long time that plants absorb sunlight in order to get energy and stay alive, but today I'll be diving a little more into the science side of that explanation and taking a more in-depth look at the chemistry and physical processes which take place in photosynthesis. The information for this presentation is coming out of the textbook Leninger's Principles of Biochemistry, the 7th edition of that book. Now, as humans, we all really enjoy eating food and breathing air. They're some of the things that we do most frequently. We breathe in oxygen and intake energy such as glucose in chemical forms. As a result, the body releases the major products as water and carbon dioxide when we breathe. Interestingly, plants do the reverse of this reaction. They absorb carbon dioxide and water, and when light is provided, they produce fresh oxygen and other carbon-based compounds which are used in the plants for various purposes. This process for plants is called photosynthesis, the production of chemical compounds using light. In 1937, a man by the name of Robert Hill performed an experiment where he took leaf extract, added a special chemical called a Hill reagent, which changed color when it received electrons. What he noticed was that the color of the reagent did not change when there was no light being shown over the leaves, but when the leaf extract was illuminated, he observed oxygen being produced, and the reagent changed from a blue color to being colorless. This showed that there must be some process in plant cells which require light to produce oxygen and allow plants to live. And this later became to be known as photosynthesis. Now, photosynthesis happens in two smaller subsections. Firstly, there's the light-dependent chemical reactions which form highly energetic molecules in the plant cell, such as ATP and NADPH, which are then used to convert carbon dioxide to useful energy molecules such as sucrose and starches for transporting energy and storing it, respectively. But how do plants absorb light, and why can't we humans do that? It would be really convenient to just sit outside soaking up the sun instead of having to pay for pizza deliveries. Unfortunately, this considerable disadvantage that we humans have is caused by the way that our cells are made, specifically in the mitochondria. Human cells use a process known as oxidative phosphorylation, which is how we get most of our ATP. In oxidative phosphorylation, molecules such as NADH are oxidized, meaning they lose electrons and hydrogens, and oxygen accepts them, creating water and generating ATP in the process. Plants function in a very similar way, and yet quite differently. Photosynthesis happens in a special organelle inside plant cells, the chloroplast. It has the same kind of enzymes which make ATP and transport electrons. However, in many ways it is flipped inside out and backwards compared to the human process of energy production. For example, instead of accepting electrons, water donates electrons to NADP+, releasing oxygen in the process. Also, when there is an electrochemical gradient in human cells, the gradient is flipped inside out for chloroplasts. As such, ATP is synthesized outside the chloroplast rather than inside of it. Chloroplasts are part of a family of organelles in plants called plastids. Plastids can self-reproduce, they have double membranes, and they can even be converted to other types of plastids. plant cells that don't do much photosynthesis can adapt to have a myeloplast rather than chloroplasts, which store energy better than chloroplasts do. Amyloplasts are found often in the roots of plants, which makes sense because what would they do with all of the machinery for using light energy when they're underground? Okay, so we've determined what is happening in photosynthesis, and we know where it happens, but how does light allow for these chemical reactions to happen? I mean, light isn't a particle with mass like electrons and protons and atoms, so how can it possibly interact with them in a physical way? Well, it kind of is a particle, but we won't get into that right now. You know how stepping outside on a sunny day is warm? That's because light contains energy which can warm you up. On a molecular level, we usually talk about light in terms of photons, small packets of light which have a certain energy, which we can define with this equation. The term lambda refers to the wavelength of the light. Light wavelengths determine the energy which a certain photon might contain. When light makes contact with an electron in an atom, that electron gets energized and reaches what is called an excited state. However, the excited state is unstable and goes away quickly, releasing another photon of energy. In plants, certain portions of chlorophylls, the pigment which gives plants its green color, absorb light into an excited state. But instead of releasing another full photon of energy, some of the energy is kept and transferred to another molecule nearby, which can then be passed along for more energy. Plants usually contain two main types of chlorophyll, types A and B. These two chlorophylls work together to take in most wavelengths of visible light, except for the color green, which is what we see reflected to us. In addition to the chlorophylls, plants contain pigments called carotenoids, which help to absorb more wavelengths of light than the chlorophylls alone can. The most important ones are called beta-carotene and lutein, which absorb light in the red, yellow areas of the light spectrum. Now, these pigments aren't just randomly contained in chloroplasts. They're arranged in such a way so as to transfer the energy they get from light to a photochemical reaction center. These arrangements are called photosystems, aptly named. Some pigments in the photosystem directly link to the reaction center to transfer energy. Others function as antenna molecules, which transmit the energy from a further distance. Once the energy reaches the reaction center, electrons in the reaction center get excited, and they create an electrical current as molecules are oxidized and reduced. These reactions form a charge separation in the chloroplast, and this is how the electron transport chain of plants gets activated, and therefore how ATP is generated. To learn more about how these photochemical reaction centers work, we turn our attention next to bacteria rather than to plants. In bacteria, we see two types of reaction centers. The first reaction center type is called the pheophytin-quinone reaction center, or the type 2 reaction center. This center is a pigment which gets excited and transfers electrons to a cytochrome complex, an enzyme which transfers electron into the electron transport chain. The chain creates an electrochemical gradient, just like in human cells, leading to ATP synthesis. The electrons are transferred from the reaction center pigment to another pigment called pheophytin, and then from there to the electron-carrying molecule quinone, and finally to the cytochrome complex. Once the electrons are transferred to the cytochrome complex, another cytochrome brings electrons back to the pigment so that it is ready to collect energy once again. This creates a cyclic pathway in which the reaction center can be excited and create an electrical gradient over and over again. The second type of reaction center, type 1, is called the iron-sulfur reaction center. In this type, the reaction center, after being excited by photons, gives off electrons to quinone, which are then given to a cytochrome complex. From there is the same pathway of creating an electrochemical gradient as in the type 2 reaction center. However, in addition, this type of reaction center also allows some electrons to be transported to ferredoxin, an iron sulfur protein, which then creates NADH, another energy molecule. You might think that it would be difficult for chloroplasts to keep all the energy they collect from the sun. You would think that some of it would get lost as it moves towards the reaction center. However, this is not the case. In fact, the pigments and proteins in chloroplasts are oriented and held in such specific arrangements that 90% of the energy which gets absorbed is maintained all the way to the reaction center. Alright, now we're going to turn our attention back to plants instead of bacteria. In plants, there are again two types of reaction centers, and they work together in order to get the most energy. As we talked about before, they are called photosystems, and there are two types, photosystem 1 and photosystem 2. Photosystem 2 resembles the pheophytin-quinone type in bacteria, and photosystem 1 resembles the iron-sulfur system. All plants have both, and without both, plants could not produce oxygen that we breathe. Instead of being cyclic, photosystem 2 receives electrons at the reaction center from an oxygen-evolving complex, which reduces water to give electrons. Then, once light is added, the pigment is excited and releases electrons to pheophytin, which this time gives the electron to what is called a plastoquinone. These electrons are then passed to another cytochrome complex, which makes the electrochemical gradient just as before. Photosystem 1 receives electrons from the cytochrome complex where the electrons from photosystem 2 got left at. Once its pigment is excited by light, it gives off electrons to a series of chlorophyll A molecules, which then give the electrons to an iron-sulfur complex, and finally once again to ferredoxin, which this time produces NADPH rather than NADH. These two photosystems are linked together by the cytochrome complex, which creates the proton gradient, as well as transfers electrons to the pigment of photosystem 1. The electrons are recycled as they flow in a cyclic path. This is referred to as cyclic photophosphorylation. The last part of this section of the book talks about how plants adapt to changing light conditions. When there is not as much light, say in the winter, plants need to adapt in order to stay alive when not as much light is available. This change is accomplished by something called the light harvesting complex, which changes its conformation between two states based on the needs of the plant cell. In stage 1, the light harvesting complex is in position where it can harvest as much light as possible and use it to directly produce energy instead of recycling it. In stage 2, the complex allows for cyclic electron transfer when there is plentiful light to harvest from. Okay, so what have we talked about? We learned about chloroplasts being the organelle, which gives plants the ability to photosynthesize. We've learned about how pigments in chloroplasts capture light energy. We've learned that these pigments transfer energy to reaction centers in what are called photosystems, and we've learned that plants have two types of photosystems, types 1 and 2. We've seen a little bit about how these systems work together to create energy in the cell, and we've introduced the idea of how plant cells adapt to changing light conditions. And that's all I have for you today on photosynthesis. I hope you enjoyed this video. Have a wonderful day.